Hello, good evening or good morning or good afternoon, and welcome back to our first OpenFlow session for a while. We've been away for a while, but we are now back. And today we're going to be looking at phenotyping together with cell cycle analysis. Now, just a couple of housekeeping things at the beginning. Hopefully you saw on that splash screen when you entered, there are a couple of ways of getting in touch with us. If you've got any questions, it's best to put those into the Q&A tab, and we'll answer those. We'll either stop and answer those, or we might answer them via text, or we might answer them at the end of the session, but we will try all of your questions. You can also use the chat function if you just want to say hello, or if there are any issues with the audio or the video. So who are we? So my name's Derek Davis, and I am the Science Technology Platform Training Lead at the Francis Crick Institute here in London, and about 3,000 miles away in New York. I'm joined by my colleagues, so I'll hand over to them to introduce themselves. Kathy?
Okay. All right. Thank you so much, Derek, and hi, everyone. It's really nice to be here in New York. So I'm Kathy Daniels, an ISAC SRO emerging leader, and with my wonderful colleagues, both virtually and in person. Really excited to be here for this session today and talking with you all about the Aurora. And I'll pass it to Rui.
Thank you, Kathy. Hi, everyone. Yes, we're all very excited to be here once again. And actually, this time we have some special guests. I want to introduce Anna Longini, our scientific manager here at the Sloan Kettering, and here with me is Barbara Oliveira. Also works in our lab here at Memorial Sloan Kettering Cancer Center, and I'm the director of the lab, in case some of you don't know. And I just want to mention a shout out to Barbara and Anna for all the help that they provided preparing samples, and so what you're going to see today is their work. So we're really excited to have them involved, and hopefully they'll be more involved in the future.
Thank you.
Okay, thank you, everyone. So what we're going to do is follow our sort of tried and tested, I suppose, route into these OpenFlow sessions. We're going to talk initially about the considerations we need for any experiment that we're going to do. So I'm going to give you a little introduction into that. Then we're going to be using the Cytek Aurora today to run some samples. So we'll hand over to Kathy, who will walk us through how we set the machine up and how we set the experimental template up. And then at the end of the session, we will do some data analysis with Rui using FlowJo. So, what do we need to consider for any flow cytometry experiment? Well, we're always going to have those two main weapons that we have in our armory, the cytometer and the fluorescent reagents that we're going to use. And in fact, the cytometer is quite often the rate limiting step. If you're lucky enough to work in a large core facility or have access to a large core facility, you're likely to have a whole range of cytometers. You'll probably be able to cover 99% plus of the experiments you want to do. But not everybody is that lucky, of course. You may be in an impoverished institute, you may be in a small startup company. So always the cytometer is going to be your first port of call. It manages your expectations of the type and how complex the experiment can be. Of course, we also have to prepare our samples. We know that flow cytometry is a single cell technique, so we need to make sure that we can prepare our samples to give ourselves a single cell suspension. And then we're going to use fluorescent reagents, fluorochromes, to help us define the population of cells that we're interested in. So understanding something about those reagents, whether they're fluorochrome labeled antibodies, as we will be using today, or whether they're fluorescent probes, which we'll also be using today, the more we understand about our reagents, the better we're going to be able to design experiments, and the better we will be able to troubleshoot them when things go wrong. We're also going to need to stain our cells or have a staining protocol, and we may also need to think about whether we need to fix our cells. And that's something that we're going to be doing today, because in order to look at the DNA content in our cells, we need to make them permeable in some way, and fixation is going to allow us to do that. As I said, we're going to look about how we set up the cytometer. So with the full spectrum cytometer, the Aurora, we're not using a PMT-based system. We're using an APD, avalanche photodiode-based system, but we still have to set an appropriate gain for us to be able to see our populations. And of course, we're also going to have to analyze that data. Often when we're running our samples, we'll get an idea of whether our experiment has worked or not. But of course, we need to be more specific than that. We need to make sure we're, one, looking at single live cells, and also making sure we're getting the data that will answer the question that we've asked. And we'll talk about the question we're asking in a second. But first of all, the machine that we're going to be using today, we're going to be using a 5-laser Aurora cytometer. It looks a little bit like that. So what we always need to know are the light sources that we have in our flow cytometer. And you can see from the list here that we have 5 lasers: UV, violet, blue, yellow, green, and red. Of course, we shouldn't be talking about colors. That doesn't really mean anything. We should more specifically be talking about the actual wavelength of the laser, which are all written here. But even seasoned cytometrists will fall into this trap of talking about the blue laser, because it's a lot easier to say than the 488 nanometer laser. So one difference, as we saw when we've looked at full spectrum analysis before, is that we don't have one detector per fluorochrome, but we have a range of detectors for each laser line. So the numbers here on the right-hand column represent the number of detection channels that we have for each of those lasers. So 16 for the UV and the violet, 14 for the blue, 10 for the yellow, green, and 8 for the red. As you can see, that number goes down as we move up the electromagnetic spectrum. And that's simply because if I'm using a red laser to excite my fluorochromes, I've got much less space in the electromagnetic spectrum to detect emitted fluorescence than I have if I'm using a UV laser. So with our full spectrum analyzers, we need to maybe look at our data in a slightly different way. So you'll be seeing a few of these today, these sort of ribbon plots. I'm sure you can't read the x-axis there, but this gives the laser and the number of channels, detection channels that each has. So here's our violet laser, our ultraviolet laser, then our violet laser, blue, yellow, green, and red. And as you can see, there's fewer channels in the red than there is in the UV. So what are we going to run today? We're going to run a very simple experiment, and this biology may not matter to you, may be irrelevant to what you're doing, but we have to have a way, obviously, of running this experiment. So what we've taken is peripheral blood mononuclear cells, and we're going to be labeling the T lymphocytes with a CD3 antibody, and that antibody is, in this case, conjugated to PE. We're then going to be measuring a subset of those T lymphocytes, the T helper cells, with a CD4 antibody that's labeled with FITC. So we're going to be able to look at least 2 different populations there. But we also want to look at the DNA content of these cells. So we're going to use a DNA binding dye, and in this case, we're using a dye called DAPI, which is going to quantitate the DNA that's in our cells. We know that in mammalian cells, we have to double our genome in order to divide. And we can detect that doubling or that synthesizing of DNA with a DNA dye that binds in proportion to the amount of DNA that's in the cell. So we can separate cells that are in G2 or mitosis because they will have twice as much dye as a cell that is in G1, G0 of the cell cycle. A word of warning here, of course, is because these are peripheral blood mononuclear cells, they're very unlikely to be proliferative. We're probably just going to see G1 or actually G0 population because these cells are relatively quiescent. And in any flow cytometry experiment, remember, we need to remove dead cells. So in this case, we're going to use a viability dye, and this is a dye called ViaRed, which comes from Cytek. So whenever we're starting in cytometry, and whenever we do a cytometry experiment with a dye that maybe we're unfamiliar with, we're going to go to one of these, a spectral viewer. This is a typical spectral viewer. I think this is BD one actually. for fluorescein. And we can see we've got a dotted line, which is the excitation spectrum of our dye, and a filled line, which is the emission spectrum of our dye. And this tells me that fluorescein is optimally excited at around about 495 nanometers. But we have our blue 488 nanometer laser in our cytometer. So this is where I would normally excite it, optimally excite fluorescein. And fluorescein will emit light over a range of wavelengths, but in a conventional flow cytometer, we are not going to measure all of the emitted fluorescence. We're going to have a bandpass filter that allows us to capture the peak of the emission of that dye. But that dotted line, that excitation profile, tells us that actually, if we have a violet laser in our flow cytometer, this will also excite fluorescein, not as efficiently. I might need 20 photons to hit FITC before I get an emitted photon, but nonetheless, it will excite it. And the emission of FITC is always going to be in the same region, no matter which wavelength of light excites it. So with full spectrum analysis, we actually take advantage of this. We know that our fluorochromes will be excited suboptimally by all the lasers that are not the optimal laser. So which is why we have these sort of ribbon plots. So again, this is FITC, and we can see that FITC emits in a sort of green area in the blue detection channels, but we can see that we get some excitation by the violet laser. We get a little bit of excitation even by the UV laser. So this sort of helps us manage expectations, where we expect to see photons from this particular dye. And we can look at all of these. We can look at PE. So PE optimally excited by a yellow-green laser. So that's where we're seeing our peak emission. But we also see excitation and then emission in the blue laser, the violet laser, and again, even a little bit by the ultraviolet laser. DAPI, our DNA dye, optimally excited by a UV laser, but we also see some excitation with a violet laser. Very little, if not none, from the blue, the yellow, green, and the red laser. Now, all those three dyes I'm perfectly happy with. I use them a lot, actually via red. I never used that before, so going to the spectral viewer, the Cytek spectral viewer, has actually helped me, because now I know that it's excited by the red laser, and I can see where its maximum emission is going to be. But I can also see that I get a little bit of excitation and emission from the yellow-green laser. And if I then overlay all of those four colors, fluorochromes, I can start getting an idea of where I might need to think about spillover, think about overlap. Remember, in any flow experiment, because of the broad emission spectra of our dyes, we see overlap, and we have to account for that either by compensation on a conventional flow cytometer or by spectral unmixing in our full spectrum cytometer like the Aurora. So I can see where there's going to be some overlap with the PE and the FITC. We're going to see some overlap with three of the dyes here, maybe a very little bit here as well. So that helps when I'm setting the machine up. So knowing the excitation and the emission spectra of our dyes is a good first point. Rui, you've come on screen. Is there a question?
Yes. Just before you go on, there was a question by Sadhana, who's asking, why do we need to exclude the dead cells if we don't intend to perform any culturing after?
Okay, so I'll come to that in a couple of slides' time, but dead cells will cause us problems even in our analytical experiments, because they change metabolically, so they change their fluorescence. Their autofluorescence level will change. And because they become permeable, they can take up antibodies and probes non-specifically. So we might see some non-specific binding, and that could be a particular problem if we're actually looking for just a small shift. We want to be sure that's real and not just due to a dead cell. So we should always target those dead cells. And there are ways of doing it, which I'll come to in a couple of slides' time.
Exactly. So basically, we want to reduce the amount of false positives in our analysis, right?
Absolutely.
Thank you.
So as well as knowing something about the excitation and the emission of our dyes, we actually need to know a little bit about the chemistry of the dyes as well, because this helps us think about the design of our experiment and potentially where things could go wrong. So if we look over here at fluorescein, so we're going to be using a fluorescein labeled antibody. Fluorescein is a very small molecule. It's only about 400 Dalton's molecular weight. And you can see that it's a cyclic ring-based compound. Now, if I've got a small molecule like FITC and I've got an antibody, I mean, antibody is a big protein. It's about 240 kilodaltons, maybe a bit more. If I've got a small molecule like FITC, it means that I can add more than one fluorescein molecule to my antibody. I might be able to add 4, 6, 8, 10. The more I add, within reason, the relatively brighter that antibody becomes. Also, if I'm using a small marker, it means that if I'm looking for something inside the cell, inside the cytoplasm or inside the nucleus, I may find it easier to get an antibody labeled with fluorescein into the cell because fluorescein is a small molecule. Also, small molecules like fluorescein are less likely to be affected by fixation because fixation always chemically alters structures. If there's not much of that structure to alter, it means that I may get away with fixing my fluorescein labeled cells and not losing any of my fluorescence, which is something that we have to be aware of with PE, phycoerythrin, the other fluorochrome we're using to define our cells. As you can see, PE is a big protein. You can see from its molecular weight here and from its sort of structure. And if I'm doing antibody labeling, I can really only add one PE molecule to my antibody, and it almost doubles the size of the antibody. So it means that I've got to fight harder to get that into the cell if I'm looking for something intracellularly or intranuclearly. Very difficult word to say. However, it doesn't mean to say I'm not going to use PE. It just means that knowing that it's a big molecule means that again manages my expectations of my sample preparation. And with PE, it's not the whole molecule that becomes fluorescent when we shine light on it. It's just certain pockets of that protein, the so-called fluorophores, or sometimes people call them chromophores, within there. And maybe there's 15 or 20 different parts of this PE molecule that become fluorescent when we excite them with a light source. But because PE is a protein, we have to be a little bit more careful when we're using fixatives. Because remember, fixatives will change the structure of these proteins. So in a worst case scenario, we might lose our fluorescence completely. Even in the best case scenario, we might see a reduction in the staining. So that may or may not matter to us. If we're looking for a very highly expressed marker that's either there or it isn't, something like CD4 or CD3, then that may not matter. But if we're looking for a weakly expressed marker where losing fluorescence would become important to us, then that is going to be a consideration. Here's DAPI. We call it DAPI because its chemical name is a bit longer. So we reduced that to DAPI. And as you can see, it's a small molecule, very low molecular weight. And pretty much all the DNA binding dyes that we use in flow cytometry are very small because they either have to intercalate into the helix or fit into the groove of the DNA in some way. So they tend to be pretty small. You notice there's a black box here. I couldn't find the chemical structure of via dye, probably because it's a proprietary dye. But I did find out its molecular weight. So again, it's not huge. I suspect it's a few more cyclic rings than FITC is. So again, a relatively small dye. And sometimes these dyes are called fixable dyes, and we'll come on to that in a couple of slides' time. So another question we need to ask in any experiment is how bright our dyes are going to be. And we're using PE and FITC. And you may know, and those of us who've done flow for a long time know that PE is one of our brighter dyes. FITC is a more moderate dye. And we discussed in one of our previous OpenFlow sessions how we can assess brightness of dyes. So if we look at CD4 labeled with FITC, CD4 labeled with PE here, we get the same pattern of expression in both of those plots. But with the PE marker, the PE positives are much further away from the negatives than the FITC positives are from the negatives. So this tells us that PE is a brighter dye than FITC. And we can derive a number that shows us that, this thing called the stain index. We won't necessarily go into it, but it looks at the difference between the middle of the positive and the negative peaks. But because different fluorochromes emit light in different areas of the electromagnetic spectrum, we also look at the spread of the negative population here. But anyway, we end up with a number. The bigger the number, the relatively brighter the dye. And this helps us a lot when we're designing multicolor experiments, because if we're looking for weakly expressed markers, we're likely to use the brighter dyes for us to be able to see them. Dead cells. So we've already mentioned dead cells a couple of times, and it's important to remove these for the reasons I mentioned, because they become more autofluorescent. They take up dyes and antibodies non-specifically, and the reason they take up those dyes and antibodies non-specifically is because they become permeable to those dyes. Our cytometric definition of cell death, if you like, is the fact that our cells have a compromised membrane. So although we can't physically remove the dead cells from the tube, there are always going to be some dead cells in there. We can target them, okay? And we target them in two different ways. One way would be to use a DNA binding dye like DAPI or propylidomide. Because my live cell has an intact membrane, that dye will try to get in, but it won't be able to. Whereas in a dead cell, the dye will pass through that permeabilized membrane, it'll go to the nucleus, it'll bind to the DNA, and it will fluoresce. So we can separate our dead cells here from our live cells. However, in this particular experiment today, we want to quantitate the DNA. In order to do that, we're going to permeabilize our cells. We're going to fix them. And fixation would, of course, make these live cells also permeable. So I couldn't use a DNA dye in this case because everything would be positive. So in this particular experiment, we're going to use a different approach. We're going to use these protein binding dyes or amine binding dyes, or sometimes called live dead fixable dyes, or BioLegend, for example, call them zombie dyes. They'll come up with some name that indicates these things are dead, but they work on the same basic principle. At live cells, we might get a little bit of cytoplasmic staining because, of course, cells are expressing proteins. But with our dead cells, because they're permeable, those dyes will go in, and now this time they don't bind to the DNA, but they bind to all of the proteins in the cytoplasm of the cell. So we can separate our cells which are dead from cells which are alive. But the advantage these dyes have is we can now fix them, and that fixation locks that staining in place. So I know that these cells were dead before I fixed my sample. Now, post-fixation, they're all dead, but I know before I fixed them, these ones took up that viability dye and these ones didn't. So today we are going to be fixing our cells, and fixation, why do we want to do that? Because fixation locks everything in place, doesn't it? It stops the cell, gives us a snapshot of that cell at a particular point. It means that if we want to look for a metabolic function of the cell, of course, that's possibly not so useful to us because we need our cells to be alive in that case. But actually today we don't. We're looking at the expression of two markers and we're looking at the DNA content. So fixation is something that we want to do. It will also prevent infection. Sometimes we're running infectious cells. It is convenient because if we can fix our samples and run them all at one time. And fixation will also help permeabilize our cells. And again, we have covered fixation in some of our previous sessions, but the two principal types of fixative we're going to use are either alcohols or aldehydes. Now, by alcohol, I generally mean ethanol, occasionally methanol. And here's a chemical structure for you, for those of you who like that sort of thing. Alcohols are dehydrating fixatives. They remove water from our cell. They also dissolve lipids, which is why they're very good permeabilizing reagents. But a downside for us is that they're going to change your protein structure. Now, if that's something that's important to you, we might have to think, is this the best way to go? Because if we're looking for an antibody, an antibody binding to an antigen, a protein antigen, if that is conformationally changed, our antibody may no longer bind. And remember, many of our fluorochromes are protein-based. I mentioned PE because we're using it. APC, alpha-ficocyanid, PERC-P, these are also protein dyes, and all of their tandems are also protein dyes, and they may well be affected by ethanol fixation. So we have to decide, is that what we want to do? And if we don't, then we would generally use a formaldehyde fixation. And formaldehyde is a cross-linking fixative, so it locks the structure of proteins in place. So that means that our PE fluorescence or a GFP fluorescence is still likely to stay in place. Word of warning with fixatives is that they all increase the autofluorescence of our cells as well. So that may be something that, that we need to think about in our sample preparation. And which fixative you use may depend on the application you're using. So normally if I'm doing DNA analysis, I will use an ethanol fixation. Doesn't mean to say I can't use an aldehyde fixation, but I need to know what effect that would have. The effect that it has is that the staining isn't quite as even if I'm using an aldehyde fixation. So here's a typical DNA profile, cells in G1, cells in S, and cells in G2. Now, ideally, all my cells in G1 should have the same amount of DNA, but we always see this data spread for multiple reasons, and we always try and keep that to a minimum. Alcohol fixation will help with that. So I'll have a very low CV, coefficient of variation, indicating there's very little data spread there. With formaldehyde, although I can still see a DNA profile, there's a little bit more data spread. The CV is bigger. So it's always a balance, you know, which is most important to me in the experiment. Is it being able to delineate a particular subset of cells, in which case we might prioritize our phenotypic staining, or is it to get a very accurate assessment of how many cells are in G1S and G2, in which case I might prioritize my DNA profile. And that's up to you. Now, this is something that you have to decide, remember, because you are always in control of your experiment. So what do we need today when we come to our cytometer? Well, of course, we need controls, don't we? We always need controls. We're going to need an unstained sample, which is going to allow us to see the level of autofluorescence. And I will always run an unstained sample, especially if I'm running a cell type that I'm maybe unfamiliar with. Because we're doing unmixing or compensation, we're going to need our single color controls, which are going to allow us to see where the photons that we're not interested in or where we are interested in, see where they end up in channels where we don't expect them. We might need some fluorescence -1 controls, FMOs, which we've talked about before. Actually, today for the sake of brevity, we've removed those. These are very useful when we have a continuous expression, but we know with CD3 and CD4, it should be fairly discrete. And this is all to set up our experiments. And then, of course, we're going to be running our fully stained samples. So the way we set the machine up and the way we do the analysis, again, we're in control of this. And both Kathy and Rui may do this in a slightly different way to the cartoon I've put here. So maybe the first thing we would do is remove our dead cells, because we're not interested in those. They're going to confound our results. So having selected our live cells, we may then want to remove doublets. Remember, a flow cytometry is not perfect. Sometimes cells will be in this laser beam at the same time and be measured as a single event. We want to be able to exclude that, and we'll show you how we do that. We may put a scatter gate in, and then we would look at our specific staining. So in this experiment, we want to define a population that's CD3 and CD4 positive, maybe a population that's CD3 positive but CD4 negative, and there may be some cells down here that are CD3 and CD4 negative. And then within each of those populations, we can look at the DNA profile. This question about fixation, I'll come to that at the end of this before I hand over to Kathy. That's okay. So what have Anna and Barbara done for us today? They've taken some frozen PBMCs, washed them, resuspended them back into a buffer containing a bit of protein, in this case, 2% fetal bovine serum. Cells always like a little bit of protein. It keeps them happy. It also helps keep them in single cell suspension. Then, having put those cells into 6 mils, they've divided them into our 6 tubes, in this case, our unstained sample, our 4 single color controls, and then our fully stained sample. Because we try to make these sessions as practical as possible, we've put the protocol here, which obviously you can look at this again. This session is recorded and will be put on our YouTube channel. But in brief, what we've done is add the viability dye. It's always with these fixable dyes, we will always do that first before we do any of our surface staining. We wash those cells we spun them and wash them to get rid of the unbound dye. The spinning step is quite important. We don't want to lose cells, and every time we spin, we tend to lose a few cells. The dead and maybe apoptotic cells tend not to be, they're a bit more buoyant, so they don't spin down as well. So we have to be a little bit careful that we don't want to lose any cells. Sorry, I'm moving my cursor wildly there. We're then adding the antibodies for the surface staining and incubating them at 20 minutes. Normally we'll do this at 4 degrees C to try and keep those cells sort of alive, but not really becoming particularly metabolically active. So it prevents sort of internalization of our surface markers. Again, we'll wash, spin these cells, and then we've resuspended them in our fixative, 70% ethanol. I'm not a big fan of having vortexes by cytometers, but I do use a vortex when I fix in ethanol. I take my cell pellet, put it on my vortex, and add as quickly as possible, maybe a mil of 70% ethanol. So my cells are being vortexed as the fixative is hitting them. This helps with making the fixation even. It helps with clumping, cell clumping, and it helps with our CV as well, keeping that as narrow as possible. We then put our cells into the fridge for at least 30 minutes. So here we've done it for an hour, washed the ethanol off, resuspended it in our flow buffer, and added DAPI. It's a final concentration ready. We've got 100 microliters. We're adding 1 microliter, 100 micrograms per mil, which is final concentration 1 microgram per mil, which is maybe a little bit lower than I would normally use, but there's a reason for that. And we'll look at that when Kathy comes to run this sample in a minute. With my DNA binding dyes, I always incubate them for a while. In fact, overnight incubation is sometimes the best way of doing it. It means you can do the staining late afternoon, put it in the fridge, and come back tomorrow. But here we've just done this for 30 minutes. So there's a question here about what's the minimum amount of paraformaldehyde or formaldehyde that can be used to fix cells. Well, I think it sort of does depend on the cells and the cell type you're using. I'd normally recommend somewhere between 0.5 and 4% formaldehyde. Obviously, the higher the concentration, the more fixed, if you like, they tend to become, and the more autofluorescent they become as well. So for most cell types, I would probably start off at around about 1% formaldehyde. And if that doesn't look right, I move either side of that and try that experiment again. It may be a little bit of trial and error. Okay, so that's the preamble. Let's go. Let's move to the Cytek Aurora. And I'm going to stop my share and I'm going to hand over to Kathy.
Perfect. Thank you so much, Derek. That was great. Let me go ahead and share my screen. And let me just make sure, can everyone see my screen and hear me okay?
We can hear you. Your screen sharing is just starting. There we go.
Okay. All right, perfect. Okay, sorry about that. It looked like Zoom crashed on me. Can we all see my screen again?
Yes.
Okay, perfect. Sorry about that, everyone. So we're now at the Aurora and we have an understanding of the experiment that we're about to run, thanks to Derek just giving us the outline. And I'm in front of the instrument, sharing the screen. And what you see is that we're signed in. So this is the home screen that we're going to see on the Aurora. I'm going to go over the basics of setting up an experiment and everything like that, but I might not go into really minute details because we do have prior sessions on setting up an experiment in the Aurora and troubleshooting experiments in the Aurora on our YouTube channel, but we'll still get an idea of how everything is working. So right here under the acquisition tab, what we see is we see some different options. We can set up a default experiment where it just lets us go ahead and get started. We can work from a template, we can import experiments, we can open up prior experiments. But what I'm going to do is I'm going to go ahead and I'm going to select a new experiment. By doing that, it's going to let us input all the details about our experiment into SpectroFlow and that software. Okay, so I like it. It's very user-friendly and it lets us walk through step by step everything that we need to do to set up our experiment. So I'm going to rename this experiment. I'm going to call it cell cycle plus phenotyping or immunophenotyping. Okay, don't need to put in a description, right? There's no need to there. But what we do need to do is we need to select our fluorescent tags that are a part of our experiment. So Derek told us about why we have DAPI in there, right? That's for cell cycle. We also have our CD3PE and we have our CD4 FITSI. And the last one was our Viadai red. Now, what wound up happening in our test run was we noticed that the cells were actually so beautifully alive that in our reference control or single color control and also in our fully stained sample, we didn't see dead cells to be able to pull it out. So while that was the plan, we will not actually have a viability dye to be able to detect in this experiment. And I actually wanted to use this as a quick educational tool to everybody. So even if you expect really good viability, you still want to include that viability dye. And what you can use in lieu of cells for a reference or a single color control is beads, specifically for the type of viability dye you're going to be using if they're amine reactive. They're called amine reactive beads or arc amine beads. But what you could also do is you can do a heat kill of your cells and spike them in with live cells so that you have a nice, beautiful dead cell population for your single color. So you can still include it in the experiment. And this is just a document showing how you could do that I wanted to reference. And it's from, at the time, Affymetrix eBio. And heat killing 65C for 1 minute and then placing them on ice and doing a 1-to-1 mix with nice healthy live cells will give you a good reference control. So with that, that's the explanation for why we don't have a viability dye in this experiment that we're live running today and what you can do to make sure it's included in the future. Okay, so with that, here in the library, what I could do is start typing my specific fluorochrome of interest so that I don't have to go and try and find it under the blue laser, red laser, viability section, all of that. So I started typing DAPI and it popped right up. So I double click it and it adds that to our selection. From there, I'm going to do, oops, sorry, PE. And you see there's a lot of different options. I know based on my experience that PE is optimally excited off of the 561 nanometer or yellow-green laser. So what I'm going to do is I'm going to scroll down here and I see under yellow-green, there's PE, and I double click and add it, okay? And then last, we're going to have FITSI. So I type that in, I see it right here. And we have our fluorochromes selected for our experiment, okay? One thing that we can go over later if we have some time is how to add in a fluorochrome in case we don't see it in our library. We could always do that, but luckily for us, these are common fluors. They're here in the SpectroFlow software already, but it's really nice and easy to go ahead and add in additional fluorescent tags if necessary. Okay, and just remember for this session as I'm going through, if anything's unclear, if anyone has any questions, please pop that question in the Q&A and Rui or Derek will interrupt me with those questions when appropriate. So now that we have our tags, I'm going to go ahead and select next. I am going ahead and running these in tube mode. So you can see in the carrier type, we have the different options, flat, V, U-bottom, or manual. I'm going to leave manual because that's the method I'm using today. And then the next thing I'm going to do is I'm going to add a group, okay? When I add that group, oops, sorry, that's our experimental sample, right? Our experimental sample set. But you might ask, well, what about your single color controls? That's a great question. And that's why we had to add in our reference group. So we have our experimental, right? We also need to make sure that we have our reference group. So that's what I just clicked on right there. And what we can do at this point is make sure that we have the correct fluorescent tags, which we do. Make sure the control type is appropriately labeled, right? So if I had FITSI, for example, on beads, I could identify that here. But we have all cellular single color controls today. And what we can also do is we can set our negative control. So if I left this blank, I would put a gate on all of my negative populations that showed up for each single color control. But what I really like doing is I like selecting our unstained sample as the negative control, as long as it's the same cell type treated in the same way as your reference controls, right? So what's really important for conventional flow cytometry and also for full spectrum flow cytometry is making sure when we're running our single color controls that the negative is matched with the autofluorescence of our single color, right? So if you're running a bead population, you need to make sure that you're not referencing a cellular unstained for that, because the autofluorescence is much different. And also here for these single color controls that we have that are cellular based, I like to reference that unstained cell sample, because when you stain your cells, there might be some background of your negative population due to non-specific binding. And then it's no longer a true negative, right? Because it has background fluorescence from that fluorochrome. So for me, what I always suggest is have that unstained that's treated in the same way, so the autofluorescence is matched, and make sure that you have an unstained negative control. If you have multiple cell types, say you had it to, that we're using PBMCs that were thawed today, but I needed to take a cell line to have a nice positive control, I could define an additional negative control and say cell line negative control, right? And then here, I could say if that's for PE, that's my appropriate negative control if it's a different cell type. So we don't need that today, but I just wanted to make sure I called that out so that everyone's aware of the best practices. Okay. All right, so I'm going to go ahead and hit save. And now you see we have these tubes with the little R's next to them, and those are our reference controls. Okay. I'm going to go ahead and select next. And here's the point where we can go ahead and add marker names. So DAPI, if I wanted, I could say cell cycle. Typically, I leave it as DAPI, but just to show you how this works, it doesn't add it for your reference group controls. It only adds it for your experimental samples, okay? And then for FITSI, that's going to be our CD4. And PE, that's going to be our CD3, okay? Now, if I had different groups and I had different panels that I was running within, I could just make sure that I only added those markers in that specific folder, and it would apply to those specific samples under that folder. Same goes for individual tubes. I can change individual markers there as well. Okay. Keywords is something that's not going to apply to us today. That's something that people do find useful, maybe if they have specific keywords, if they're looking at specific patient samples or a specific treatment type, anything like that, you could define that in the software, but we're going to skip that for today because we do not need it. And we're going to go to acquisition. Okay. Now, at this point in acquisition, what I'm going to really focus on is making sure for my events to record that I have enough from my reference group. So you see, that's my folder right here, reference group, events to record. I want to make sure that I have enough of my positive population to be able to have accurate unmixing. For cells, I like to start out at about 20,000. Okay, the storage gate is something that we don't touch, because if I selected a storage gate, that means anything that's outside of that gate will not be recorded. Okay, and then for stopping gate, that's what I'll typically focus on. So P1 in this default raw worksheet in this specific worksheet and this instrument is a scatter gate. So what I'm going to do is I'm going to want 20,000 debris excluded events that I'm recording, and I could always modify that as needed. I'm going to come back and edit this experimental setup after, so that I'm not in the raw sheet for my experimental. But for now, we're just focusing on the single colors. So I'm going to go ahead and select save and open. It's going to take a minute, or not a minute, but it's going to take a few seconds, and then we're going to be able to go ahead and get started. Okay. This worksheet I was just playing around with a little bit before, so that's why that looks the way it does. I have it set up in a way where I'm going to put it back to standard because I shouldn't have had that mix, so I can show you exactly what I do in this scenario. So let's pretend it looked just like this when we came in. Okay. So here we are in SpectroFlow. We have the green arrow, that's the solid green next to our unstained cells. And what we can do at this point is we can load our sample to make sure our settings are correct. One of the biggest goals I hope to accomplish, one of the main goals I hope to accomplish in this session today, is educating everyone and making sure we all understand how to set the scatter gains appropriately and how to make sure that the fluorescent gains are appropriate, considering we're looking at cell cycle and immunophenotyping all together in one. So in order to do that, what I'm going to do is I'm going to load a little bit of my fully stained sample in this unstained cellular reference control, and I'm going to keep the flow rate at low because we want a nice low flow rate to keep tight CVs, and I don't want to go through too much of my sample. And what I want to do is I want to check and make sure that I have my cells on scale and my fluorescences on scale for all of my individual fluorochromes. So with that, I'm going to vortex. Any questions coming through that are good for right now? Okay.
Actually, yeah, there is one that's not actually, we haven't got that far yet, but I'll try and answer it now because I pressed the wrong button when I was doing the Q&A. Marisa is asking, can you use DAPI as a live dead dye in this experiment? Well, the answer to that is no, because the cells are going to be fixed. I mean, you can use DAPI as a live dead dye, but it has to be done on unfixed cells. So,
Exactly.
which is why that we would have used one of those fixable viable dyes today if we wanted to get rid of the dead cells.
Exactly. That's a great point. So the fixable live dead is exactly what you need to use in a scenario like this as you stain them for those to see whether or not you can pull out the intracellular amines if the membrane has been permeabilized or it's starting to, the cells are starting to die off. But now that the membrane has been permeabilized with that ethanol, we cannot use it as a viability. Okay. I see Rui there too.
Yeah, there's another question. So it's also related to fixing. So after fixing, when we add dyes such as an intracellular staining, what can we do to keep the samples not lose signal for 24 to 48 hours? I mean, at least I would say that, you know, keep them in the fridge and in the dark as much as possible.
Yeah, and be careful about the use of tandem dyes too.
Yeah.
Sorry.
Probably more than 48 hours, right? I mean, more than that, it's very unlikely that you wouldn't lose any signal, right? You wouldn't compromise any of the, you know, the resolutions you already have. Yeah.
And I just want to add on to that too. So I've been in research for most of my flow cytometry life, and now I'm in the clinical setting for my position. And what I would highly suggest is if you have to see, if you have to look at intensity of signal and look at MFIs to be able to track, you know, antigen density or anything like that, you should do some type of a validation and stability experiment post staining to make sure that you're getting all the answers that you need. Because if MFI is an important readout, you might have to stay within a fresh run or 24 hours or something like that. So based on your panel, the fluorochromes that you have in there and the biological questions that you need to answer and the setting that you're in, that could all vary.
Right. And so you create more variability if you're storing for longer periods, right?
Yeah.
So the best is always to run it immediately, right?
Yeah.
So Kathy, actually, there's a question regarding the settings. I believe in the acquisition control, someone is asking, you adjusted it to 3,000 microliters. Should we change to lower volume? Can you explain there?
Yeah.
I believe it's in the acquisition control.
Yes, that's a great question. So that's a standard setting that was there by default in SpectroFlow. I tend to not worry about that too much. If I'm in plate mode, what I'll do is I'll make sure that those volumes are appropriate so that we're not running dry. For tube mode, I'm actively looking at the volume that's there, and we've added, you know, our cell buffer so that we have enough to run. I don't have the exact volume that I've added in because we did it with a, you know, a manual pipetter. So I left that because 3,000 microliters is 3 mils. I do not have 3 mils on me, right? So it's just going to keep running and that's okay. And I'm going to usually set a stopping gate based on number of gated events. And that's my preference if I'm running in tube mode, and to visually verify that I'm not running the tube dry. And plate mode is a different story. And then you could define a stopping gate of time, of volume, or of a gated population. Okay.
Right. The important thing to note is that it will stop at the first criteria that's met. Right?
Yes.
So,
Yep.
so if you say, you know, I want to record 10,000 events, and that's only a few microliters, then it will stop even before reaching the 3,000. So,
yeah.
Yeah.
Absolutely. Thank you, Rui. Okay, so with that, what I'm going to do is I'm going to go ahead. I have my full stain sample loaded on acquisition control here. I'm going to make that a little bit bigger. I'm going to change this events to display to be about 20,000, and I might modify that. The rationale for that, before I get started, I'm going to be adjusting some settings, and I want those events to refresh a bit faster. So depending on my event rate and how much is going through, that's going to have an impact on how often those plots are refreshed. Okay, so I'm going to go ahead and I'm going to select start on low. Our user settings are our Cytek assay settings that we have as a standard. And what I'm going to focus on today is our forward and side scatter gain. Make sure our cells are on scale. So we have our events going through at about 2,000 events per second. We see this pattern. And what I'm going to do is I'm going to say, all right, maybe these are my cells, right? And when I do that, if I look at my spectral profile, everything's low, right? This is my fully stained sample. I don't expect that. So where is my staining? If I bring that over here, oh, look at that. We see some profiles that we would expect for FITSI, for PE, and absolutely for DAPI, right? So what I'm trying to do here is make sure that I'm pulling out where my cells are. Okay. You also have the capability to add in through that option here, the population hierarchy, which already existed and I just hadn't seen it. And the P1 is 26%. So that's 26% off scale. So what I want to do now is I want to come to this instrument control and go to my user settings and bring that forward scatter down. Right? The rationale for that is everything's off scale. It's too bright for forward scatter. So I'm going to try bringing that down to 300. And then I can hit restart. When I do that, you'll see, all right, not much different. Let me bring that down a little bit further. I'm going to bring that down to 100 and restart again. When I do that, you see now I only have 7% off scale, but that's including some of these. If I bring this up and over, what do I now see? I see I have 52% roughly, and this gate can be cleaned up. It can be prettified. I don't know, that's not a word at all. I just made it up for this session, but I can update this gate, and I will. But what I want to do now is I want to make sure that I'm fine tuning that forward scatter and bring it down a little bit more, maybe to about 80. I'm gonna restart. And then you can see we have our nice PBMC population, okay? I still have plenty of samples, so I know we're good there. I can still keep it running and adjusting my fluorescent gains. What I want to do though now is I want to adjust this a bit so that I'm not seeing all this debris. Remember, we're on a full spectral instrument, and what winds up happening is every single event that you acquire is going to be stored, and you're going to have big data files because we have a lot of detectors here. Believe it's somewhere around 60 some odd detectors. Rui or Derek, please correct me if you know the exact number, but I'm just estimating there. So we just want to be mindful that we're not creating data files that are too big. So now I'm going to go to the threshold, and I'm going to adjust this threshold. It's at 50,000 right now. Each tick here is about 200,000. So I'm going to change this value to 400,000. And we're going to see our frequency go from about 52% up to 96, right? And we're getting all of our populations that we need. So this looks great, okay? Now, the next thing that we need to do is make sure our gains are appropriate from a fluorescent perspective. Typically, as long as everything is on scale, you're good to go from a perspective of immunophenotyping. But we need to look at that, okay? So what you see here, I'm going to delete this quickly. What you see here in this full spectral profile is that we have signal on scale for the blue laser line and also for the yellow-green laser line. So we're good there. And the UV and the violet is where we expect to see our DAPI. All is well and good there, but we want to make sure for cell cycle, it's the appropriate settings. So you can see for UV, it's the brightest on our UV7 detector. So what I'm going to do here is I'm going to make this histogram nice and big so that we could all see it. And in the properties, I'm going to change the x-axis scale from bi-exponential to linear. Okay. And then at this point, what I'm going to do is I'm going to see if we need to reduce the detector gains for UV. So to bring the UV a little bit lower, we can hit a negative value and try that out. So if I hit -50 and hit enter and refresh, you can see where we're at in terms of our DAPI signal, right? So when you're looking at this, we want to make sure that we're in linear. This is our fully stained sample, right? So this is where our G1 peak is. Typically, when you're looking at different types of cells that will be proliferating, you would expect to see a G1 and a G2 peak. We don't have that today, and that's okay. We're just showing you the principle behind what we would do in this scenario. But now we know that everything is on scale. What you want to be mindful of, and I'm just going to go ahead and stop this run. What we want to be mindful of is we want to make sure that when we're setting up our gains for cell cycle, you understand the type of tissue that you're working with or the experiment that you have. So if I'm looking at a cell line that's proliferating, we might expect just to see a G1 and a G2. So you want to make sure your G2 is on scale, and the MFI should be roughly double. So we would see maybe a peak around 2 million and set your G1 around 1 million. Now, if you're looking at something like hepatocytes where you have 2N, 4N, 8N, in some cases even 16N with hepatocytes, I've worked with them before, what would your goal be? Make sure that your highest ploidy is on scale so that you can see the full breadth of the different populations. So you might need to reduce your UV and your violet gain to bring the signal on scale. Okay. Now, what I'm also going to do quickly is I'm just going to double check my DAPI single stain control and make sure that that one is also on scale in case there was any differences between that single stain and the fully stained sample here. Okay. So I loaded that DAPI on and I'm going to hit start again. And I'm just going to confirm these settings. Okay. Any questions coming through? We're all good. Okay, looks like we're good. Okay. All right, so now we can see the reason I did this, right? I threw on this DAPI, and what would have happened if I had ran this experiment with this single color control as my reference? It would have been off scale and the unmixing would have been incorrect rather. So you can see for this UV7, it's off scale, and I need to bring this -50 value down, maybe to -80. And restart. Okay, we can see the signals on scale, but I want to bring it down a little bit lower. So I'm going to bring this down to maybe -83, -84. Okay, and then if I come back over here, what can we see? Our UV is on scale. Now, what we need to do as well is make sure that the violet's on scale. So it's around V3 where we have our maximum signal for violet. So I'm going to right click on this plot, change it to V3, and I'm going to make sure that that's in linear as well. And we're on scale. It's okay. So for this experiment, I don't need to modify the violet, but if it was off scale, I could bring it down, right? Now, this is a little bit high still. So if I wanted to, I know that might seem a little bit silly based on what I just said, because it's at that higher end in the logarithmic scale, maybe I'll bring it down a hair. So I'm changing my mind. I'm saying, all right, maybe let me bring it down by 20%. And all of those detector gains come down an equal percentage. Okay. Now, why is this important? And why is this the approach that I'm taking to carry out this experiment? Why am I not just changing individual detector gains? The reason I'm not changing those individual detector gains is because I want to maintain the spectral profile of all of my fluorochromes. So if I start playing around with individual detector gains, that's very, very difficult to do because you're changing individual values. It's easy to get lost, and then you're changing the overall pattern of that fluorochrome and the spectral profile. Okay. I hope that makes sense to everyone. If that doesn't make sense, let me know. But this is the preferred approach, making sure everything's on scale. And if it's off scale, bringing that full, all the full channels down by a certain percentage. Okay, so I know that all of my detectors are on scale now. I know my forward and side scatter is good. And now I have to run my unstained and all my reference controls. So what I'm going to do, because I just ran a DAPI single color and a full stained with DAPI through the instrument, I need to run 10% bleach through the lines followed by water at a high flow rate to make sure that I don't have carryover, so that when I run my unstained, it's picking up that DAPI from the lines. Okay, so I'm changing the flow rate to high. I have 10% bleach on that the wonderful Feng prepared for us, and she's here in the lab, and we're very grateful that she did that. This is very much a team effort OpenFlow. So we're running that 10% bleach now. Yep.
Yeah, while you're running the bleach, there's a question here that's related to the, to adjusting the gains.
Yeah.
So actually, first I would just comment on the fact that, this is a spectral system, and I know that sometimes this can be a bit confusing for people that are still novice with spectral, but it's important to know that you should use all of the lasers that are available, right? Because the more lasers you have on, that means the more detectors, that means the more information you have to distinguish the different peaks and then to do unmixing. So it's important to adjust all the gains so that everything is in scale, because if one of the lasers is off scale, then that's going to interfere with the quality of unmixing. Now somebody's saying here, so why not apply the same percentage of change to all lasers? So if you want to answer.
Yeah, that's a great question. So if we think about it, when we looked at our fully stained sample, right? I know we're not seeing it now because I'm running the bleach and I'm actually about to put water on. But if we think about it, we have FITSI and PE in our experiment as well. Now, when I go ahead and I run those, or I ran those for our full stained, we saw that those were on scale. If I reduced the blue laser detectors all by a certain percentage and brought those down, and then I did the same to yellow green, that's going to reduce the stain index or the separation resolution of populations for our CD3 and for our CD4. Okay, so by bringing that down, yes, everything would be brought down the same frequency, but we would lose resolution of those populations. So luckily for us, the way we've designed this panel for having UV and violet is the main laser line for DAPI, and our FITSI and PE is most notably seen in our blue and yellow-green laser lines. Through that, we've designed this panel so that we'd only have to bring down the UV a specific amount or the violet a certain amount. And we don't have to touch the blue and the yellow, green, or the red, because those are not as excited, or those dyes that we see are using FITSI and PE aren't as excited by the UV and violet, and that's not our primary detector. So we want to make sure that we get the best resolution and only bring down when necessary for laser lines. I hope that answers the question sufficiently.
Yes. Thank you, Kathy. But there's another question. Actually, it's related to gating. I mean, we've addressed this in previous sessions, but if you can just answer, quickly. So someone is asking, how do you decide on the shape of the gate? Is it recommended to have a strict gate around the population or a very relaxed one?
Okay, that's another great question. All questions are great. So I think for it all, that's very, very subjective, right? Because it all depends on what we're looking at. I tend to keep a pretty broad gate for forward and side scatter most of the time because I want to exclude my debris, but I want to make sure that I'm including most all for my applications typically, because in case there is any activation in cells, the scatter profile might change. So if I kept that really tight, I might lose out on some activated cell subsets, right? By keeping it broad, I'm making sure that I don't lose those. What I would advocate for you to do in your research or whatever your work is to utilize backgating, right? Look at look at a gated population and backgate to see where they fall in all of their parent gates to see if you need to broaden it or make it more strict, right? For viability maybe, or for singlets or anything like that. So play around with your data and answer that question based on your biological question.
Right.
Okay.
Kathy, I would add to the fact that, and actually somebody was asking also before, so why are you getting also the granulocytes, or actually the monocytes, and are they important in this case? I mean, we're actually only have lymphocyte markers, but so the whole thing is we have to also distinguish between acquisition and analysis, right? Data analysis.
Yeah.
So here, when we're acquiring, we really just want to acquire the data, and then we'll do the analysis, the propagating afterwards offline or on laptop. Right here, we want to actually gate. We want to be less conservative so that we can see everything, and then we adjust all the settings appropriately so that when we acquire everything appropriately, right? But the gating itself, we can worry about that later during analysis.
Exactly. And I'll just point out that one thing I want to be mindful of is, for CD4, for example, we see that on monocytes, not just lymphocytes, right? So in this scenario, monocytes are that CD4 moderate, and the lymphocytes are going to be brighter for CD4. But if I wanted to make sure, like if I was unsure about expression of populations, if I gated in a certain area and something's off scale, I need to know about that so I could adjust my gains appropriately. So I like in this initial setup to keep things a little bit more broad to make sure I'm not going to regret my choices after the run. And just for sake of time, so we're already at 210, what I did is I ran the unstained control, and you see, I'll just go back to it now. But I just wanted to show everyone with that unstained, if I go back to it, right, by clicking on that sample, we're no longer seeing that DAPI signal. We're seeing the autofluorescent profile of these thawed PBMCs. So that bleach and water run was successful. And as more questions are coming through, we can absolutely answer them. I'm just going to start going through and recording these single color controls. And to save time for myself, I'm going to skip over that DAPI so that I don't have to run bleach and water again. And I'm going to go to FITSI. Okay, and I see that there's another question in the Q&A. Is there something that we haven't covered just yet as I'm running these single colors?
No problem. It's about fixation, so I'm typing an answer.
Oh, perfect. Perfect. Okay. All right. Now, another tool that I really like, or something that I really like doing as I'm going along, is I like checking to make sure the spectral signature makes sense. So this is our FITSI. This does make sense to me, but if you're curious as to why that makes sense, let's talk about it. So I'm going to go ahead and record. We can have an understanding of what the spectral profile of a certain fluorochrome is by looking at spectra viewers online. So I know that FITSI is excited off of the blue laser line, and it emits, believe it's right around 520. If my memory serves me correct, and that's around B1, B2, right? So when I look at that, this is what I see. Now you also see you see this kind of population right here from B1 to B3. You see a moderate, and then you see a high. So remember, I just mentioned that monocytes are going to be CD4 dim. So if I click on this icon right here, and I just right click, and I do blue, let me do blue 2. What do we see by side scatter? We see our negative. We see our monocyte that has a different scatter profile by side scatter. And then we see our CD4 lymphocyte. So that's a really cool thing to see. And we can also see the spectral profile of FITSI. So this is checking out to be what it should. Okay. And then I'm going to run PE next. I'm thinking based on how timing is going, it might be a little bit rough for us to do analysis today because I talk so much, but I think everyone's used to that. Let's throw on the PE.
I was wondering how I'm going to do analysis in 10, 15 minutes. But...
Yeah, I think we'll have enough to go over today in this session.
Right.
Okay, so I'm loading the PE now, and then DAPI will be my last, and then we're going to do unmixing, and we're going to run our sample. Okay. All right, so we see our samples are coming, our cells are coming up rather. We see our yellow-green 1 signal, nice and bright. I'm going to go ahead and I'm going to record. And going back to a little bit earlier when I ran the, when I set up the reference controls rather, what I did there was I set the negative as our unstained. If you remember, I said I want to make sure there's no background fluorescence or anything like that through non-specific binding. If you titrate your antibody, you do that to minimize that non-specific binding and make sure that you get the best separation, right? And that should mean that you don't have any shift up of the negative. But on this single color, there's a little bit more, right? So there's a shift up in the background, and that's what's so nice about having that beautiful unstained control. That's how you know you're a cytometrist. You say it's a beautiful unstained control. Okay, I'm going to add a little bit more buffer to our DAPI single color control here. Make sure we don't run out. Okay, should be good there. And then we'll get to the unmixing. Okay. And, you know, keep in mind, again, we're doing this as an educational tool and everything like that. So if we had a real true experiment that we were running for cell cycle and immunophenotyping, the cell type would be different. We might have some different populations, but this is just walking through the protocol of what we would normally do in this scenario. Okay, so I'm going to go ahead and record. We have our DAPI. Okay, and that's started. As that's going, I'm just going to point out, because this might take a minute or so to run. These user settings now have been adjusted. So what I would have the capability of doing is saving these specific user settings because they're specific for my experiment, right? And then what winds up happening with SpectroFlow and with the QC beads that you run to check the performance of the Aurora every day, it's going to adjust those gains across detectors to meet target values. And the percentage of change that's applied to all those detectors is then applied to your individual user settings when you import those in. So that's really, really nice. And it's a really good tool of the software. Okay. And another thing just to point out, I know this worksheet can be overwhelming to a lot of people, right? There's a lot of histograms here. And if you feel overwhelmed by that, just look at this full spectral profile. It's really nice, and it shows us all the detectors in one shot. So I find that to be the easiest thing to look for to really make sure that we have the signal that we expect instead of looking at the individual histograms, unless you're in a scenario like we're in today where we're adjusting gains for cell cycle where we have to look at it in linear. And another helpful tool, in case that properties window is not showing up in your SpectroFlow software, you select a plot, right click on it, and choose properties. And then you can change your parent gate and what it's showing up on, and change some visual things there as well. This is the full spectral profile, so we can't change it to linear, but you can do that on the histogram. Okay. So with that, we're now going to go through to unmixing. And we can see now, because I've run all of the reference controls for ourselves today, that the unmixing is now ungrayed out, and I can click on it, and it's going to take me to that window. No questions in the Q&A. We're doing good. Okay. So here we are. We have the appropriate stain controls. Everything's looking good. You'll see this option here, autofluorescence as a fluorescent tag. That's something that's really, really exciting about full spectrum flow cytometry. We have the capability to reduce the impact of autofluorescence by checking off autofluorescence as a fluorescent tag, and it's essentially the flavins, the NADPH, the different molecules inherent to the cells that are autofluorescent are treated as their own fluorochromes, so that the impact of those can be reduced. This is really, really helpful. Say if you're looking at alveolar macrophages, really, really highly autofluorescent or different subpopulations. And that's not the purpose of today's session, but something that's good to know. And because these are PBMCs with low autofluorescence, we do not need to do this. And I would suggest in your experiments, looking at autofluorescence as a fluorescent tag incorporated and not, and comparing results to see what gives you the best results. So I'm going to go ahead and go to next. And now here we are. We're just going to adjust our gating. So I'm going to go ahead and adjust this. And what I did here, you're going to notice that that gate all of a sudden changed for all of these. When I move this around, I selected the control key. And when I did that, it adjusted this gate for all, okay? And it's adjusting them for all cells. And I find that to be really helpful, so I don't have to spend all this time adjusting it for individual samples. And then what I'm going to do is I'm going to make sure that my positive are on the brightest peak. Here, if I were to choose the positive for our FITSI as anything that's positive, including monocytes and lymphocytes, the unmixing might not be as great. So I'm going to bring it to the brightest portion here. And you can see, you know, we're going to go to the brightest intensity, and that'll yield better unmixing results. Our PE is right here. We're looking good. Okay, so everything's looking appropriate, right? And then what I'm going to do is I'm going to go ahead and select live unmix. What that gives us the ability to do is that gives us the ability to run an experiment and see live unmixing. Now, what I want to do is I want to set up this tube so it's going to go to our default unmixed worksheet, or to set up a new unmixed worksheet so that I don't have to look at it in the raw worksheet. So I'm going to select edit on this experiment. I'm going to go to acquisition. And for my experimental group, I'm going to change that worksheet to our default unmixed worksheet. And I can change my stopping gate later on, or I could just say I want 100,000, right? Because we're running a little bit late on time, and I don't really have my gating strategy defined just yet. So I'm going to hit save and open, all events 100,000. And when I select save and open, what you're going to see here is that we're now on the unmixed, okay? And this was someone else that had made modifications to that default unmixed. So what I'm going to do is I'm going to delete all of their plots, okay? And I'm going to go ahead and I'm just going to start my own. So this is our scatter, which is fine. I have my tube, my fully stained tube here, right there. I'm going to go ahead and vortex, throw it on. We're just going to start recording, and I'll set up my gates as we're going. Okay. Now, like Derek had mentioned, we might want to start off with viability if it had worked out for this experiment, but because it didn't, that's okay. We're just starting off with scatter. Okay, so I'm going to go ahead and I'm going to record, and we're going to set up our gating. Okay. Okay. Ooh. I double click on this P1. It's going to show me a plot that already has the parent of P1. And I'm going to look at singlets from here. So I'm going to right click on this. And you're going to see you don't no longer see all of those individual detectors. And I'm going to see a forward scatter height. I'm going to select that polygon. Each click is a node. I have my singlets. And then from there, I'm going to look at my CD3 versus my CD4. And that looks really good, right? If I want to bring this down and just focus on my lymphocyte population, we don't see that moderate CD4 anymore. If I bring this up, those are CD4. We don't see any CD3, right? We just see a change in autofluorescence. So if I want to bring this down here, I have the option of doing that, right? And then like Derek mentioned, we could set these different populations. Now, you see here, this isn't the best. So I'm actually going to delete that. And I'm going to change that to a polygon because it's going to help me better choose the population that I want because it's following the pattern of the autofluorescence. And then maybe for our CD3, CD4 cells, the rectangular gate is just fine. Okay, and then from there, I'm going to add a histogram. I'm going to look at cell cycle DAPI. I'm going to right click and I'm going to go to properties and I'm going to change that to linear. Okay, and I'm going to change that so I'm looking at P3. Okay, and I'm going to do the same thing. Ooh, accidentally double click there. I'm going to duplicate this. And I'm going to set that parent as P4. Okay, and now this, like we said, we're only going to have a G1 peak because we're looking at thawed PBMCs that are not actively proliferating. But if you were looking at tissue, now what would you be able to see? Or some cell line or anything that has individual markers and something that you're looking for, we'd be able to identify based on individual populations the cell cycle profile. Okay. And just to also note, what we can do is we could always come in here, we could rename. So P3 is our CD3 T cells, P4 is our CD4 T cells. And we can play around with any of that. Okay, so I definitely abused my time and went a little bit over. I think what makes the most sense to me is to skip the analysis portion for cell cycle, since we already have analysis of cell cycle covered in FlowJo in one of our prior sessions, but I'll leave that to Derek and Rui and open it up to any questions that we might have.
Very good. Thank you, Kathy. This is wonderful. So we have
stop my sharing for now too.
Yeah, let me share my screen also, and we can start maybe looking at the analysis. And I'll try to do this in, let's say, 10-15 minutes. I mean, most of it Kathy just showed here, right? So while she was acquiring, she already went through kind of an analysis gating strategy. And so I'll basically reproduce this in FlowJo. So I'm going to open FlowJo. You know, I was planning to show a few things, but you know, for the sake of time, I'll just go ahead and show you how we would do this analysis in FlowJo very quickly. So I grabbed my folder, and here, if you see in the cell cycle folder, this is basically the data that Kathy just acquired. The only difference is that we actually acquired this data the day before yesterday, and so we have the data here already to be able to analyze. But essentially, these are the files that you would get from the Aurora. And so you notice that there's a raw and unmix folder. I'm actually, you know, there are these also experiment folders, so you can export things as FCS files, and you'll have them here, you know, grouped by the folders or by the sample groups that were added in the acquisition software. And you have the raw and the unmix, and I'll explain a little bit the difference between them. These are just files when you export the experiment. You'll notice there are these WTML. Even the experiment here folder is kind of a zip file that contains the worksheet that you build, etc. So already the gating strategy. So in case you want to re-import into the acquisition software, unfortunately that doesn't work for an analysis software like FlowJo. But you know, I'm confident enough to just drag this folder and see what happens. So you can see that FlowJo recognizes what the FCS files are and adds them into folders that correspond to the original folders that you just dragged in. So I can actually see all of the samples here, or I can just go immediately to the unmixed, and I can see all of the unmixed files. Notice that since we had subfolders within the raw and within the unmix, FlowJo also place those files within those folders. So I can do what Kathy was doing and just go directly to the unmixed. This is with ethanol, fixed with ethanol, and this is fixed with PFA. So let's try and look at both of them very quickly. So I'll start with the PFA, and again, just a brief comment on unmixed versus raw data. So the raw data is what Kathy was just acquiring when you saw those spectral plots. Basically, that spectral plot is a summary of all of the channels, the 64 fluorescent channels that Aurora has. And so you can see them individually. And actually, I can go very quickly to the raw files. And if I open this, you would see that the list of parameters are actually all of the channels, UV1, 2, 3, up to UV16, and then the V1 to 16, etc. And you would have all the channels. If you actually want to understand better about the configuration, you can go quickly to our website, fccf.mskcc.org, and then you can go to instruments, analyzers. We have our Auroras with the respective optical configurations, and you can see that these are the channels, the 64 channels that the Aurora has. So this is what corresponds to UV1, UV2. So you can see it goes from 365 nanometers to 380. So it's a 15 nanometer window. This is another 15 nanometer window, etc. So here's basically a layout of the Cytek Aurora configuration for 5 lasers, right? So when we're talking about UV1, UV2, this is what we're talking about, really. So going back to FlowJo, this is what we would be seeing, the signal in each of these channels, in the raw channels. But once we unmix, what we do when we unmix is we're calculating how much fluorescence of each fluorochrome is in each cell, right? So what we'll end up when we unmix is a file, an FCS file, with the respective fluors as channels, right? So now we no longer have the detectors displayed. So I'll show you here. So we'll be looking at the unmixed, just like Kathy did. And so I can open this file, and you'll see that this unmixed file has the fluorochromes, DAPI, FITSI, PE, APC, and no longer the raw detectors, right? So we can start with the analysis that Kathy did, or we can show, for instance, the strategy that Derek mentioned. So we can start with the Vi dye. So here it worked, right? We had a viability dye, and I forget, I think this was Ford scatter that Derek was mentioning. You can see here that, yeah, sure, we can see, so first thing, there's a lot of data on the half top part of this, the plot, and very little data or none in the bottom part, right? Sometimes what happens is when you load FCS files from any vendor instrument, FlowJo may be able to automatically recognize the scaling. Sometimes it doesn't do as good job, but it really depends on the type of files and the type of instruments and how you also scale it in your own instrument, and the resolution of the instrument, the detectors. So normally we always have to adjust a little bit the scaling. So I could come here and just adjust this scaling, and I would introduce a little bit more of or less width basis, and you can see that now we're starting to see a bit better. So I just apply here, and now we can see a little bit more of the data. And so basically what we're seeing here is the lymphocytes, the monocytes, and these are the dead cells, which most likely are dead lymphocytes, because that's the majority of the cells we have here anyway. But if you have any doubts, you can always try different approaches. So for instance, I'll do the analysis like this here. And so I'll use a polygon, just like Kathy was using, to be able to better select the population I want. I'll call these live lymphs, because I'm gating now only on the lymphs, because we're interested only in this case in seeing the lymphocytes, right? So now I double click here, and I'll do the Ford scatter. Let's do Ford scatter height versus Ford scatter area, and now we can eliminate. So anything that increases area but doesn't increase the height also, those are aggregates, right? So single cells, great. And here you could be more or less stringent. It depends really on the type of analysis that you do. It's always difficult. This is the most subjective plot I usually say in flow cytometry. It's always difficult to know when the singlets end and the doublets or the aggregates start. And so here's really a subjective call. If you want to really make sure that you have singlets, I would say be be more strict and less strict, because the more of these that you gate on, the more false positives, let's say, you'll get. So double clicking here, we can continue the analysis. So here now we're going to look at let's say CD3, or actually we had CD4. I'm trying to remember what Kathy did. CD4, and then here we could look at CD3. Do you see that this looks off scale? Now the thing is, notice here 10 to the 5th. This is the automatic scaling that FlowJo applied. But the 22-bit resolution data from Aurora actually goes to basically 4 million, something like that, 10 to the 6th, a bit over 10 to the 6th. So we can actually come here. So this data is not off scale; it's actually in scale. It's just the scaling, automatic scaling from FlowJo didn't capture that the other cells, right? The other events. So we can go again, customize. And this is the reason why FlowJo has this feature, to allow you to, from any vendor, to be able to look at your data appropriately. So here we can increase the amount of logs, positive logs, and you can see that the data will end at something like 4 million. After that, nothing is really there, right? So I click apply. And now I see the data in scale, right? So here we could gate the CD4s. These are probably the CD8s because they're CD3 positive but CD4 negative. So in the PBMC samples, but we actually have CD8s here. So this is kind of redundant. We were trying just to give, put known markers with different fluors just for the proof of concept. And so if we gate here the CD8, you can see that you would be able still to distinguish the CD8 and the CD4, right? Now, here in this case, since we don't have a beautiful separation, maybe using an FMO, like Derek mentioned in the beginning, would be appropriate to be able to set the gates accordingly. But here, for the sake of just the principle, we're going to gate on the CD4. And I'm going to use just the CD4 just as an example, and you'll see why I'm focusing more on the CD4. So let's look at the CD4 just like Kathy did. Now we're going to plot a histogram with DAPI, right? So we now go here, and you can see now that this seems very strange here, right? We have a lot of data squashed in the axis. Again, the scaling is not appropriate. It starts by automatically applying a bi-exponential scaling. We actually want to look at DAPI in linear scale, right? So we apply linear, and you still see a lot of weird histogram here, or a weird histogram. And so here we can go again, customize axis, and try to see more of the data and understand what's going on here. And as you see in linear scale, FlowJo didn't do a very good job, but again, it does already an amazing job to be able to automatically predict what is the appropriate scaling. So we can't really complain. But let's say I still see data squashed here. Do you see that it's approximating to 4 million, and there it is. Now we're already getting things off scale, right? So we could just leave it here and then apply, and you can see that this is basically the histogram that we obtain. And now you can be asking the question, well, why do we have two peaks? Well, the thing is, here in terms of staining, the fixative, there was a lot of conversation in terms of the, how much fixative, and it's not only how much should we add, also how much time should we add. In this case, we didn't optimize the protocol. So Anne and Barbara, they prepared these samples, and as you can see, what they found was that we weren't able to really stain all of the cells here. So here would be, maybe playing around with the fixative, the amount of fixative and time, also the permeabilization step. Maybe it was a problem there, or maybe a problem with not enough DAPI here to be able to stain all the cells, right? So now, if you're asking the question, well, but maybe this is G0, G1, and this is the G2 and S phase. Here it's obvious that it's not, starting with the issue here that if it was G0 or G1, the G2 should be 2 times the amount of DNA, and you can see here that this is 500, this is 1 million. So this is essentially what, 100, 2, 3, 4. So this is around 100K. So double of that should be 200K, which would be here. And we're not. We're seeing it clearly in the 800,000. So this is clearly not a related peak. This is basically debris cells without DAPI that haven't been stained. So we could conclude from here that the staining didn't go very well. And we know that the fixation with PFA and permeabilization, again, needs to be optimized. And ethanol is typically the protocol we use for DNA cell cycle, because it just gives you much better CVs. The staining is way more clear than with PFA. It's not that it wouldn't work, it could work, but you would need to optimize it, right? So let's see how it looked like with ethanol, right? And let's try, so here we go to the unmix, not the raw, and let's do the same strategy very quickly with ethanol. Here, actually, I'm going to start and I'm going to do this gating strategy in a slightly different way, so that you can see that it doesn't really matter where you start from, as long as you understand what you're doing. Is fine. You know, the important thing here is to clean out the data as much as possible and then analyze exactly what you want, what you intended to. So in this case, we're just looking at lymphocytes, we're excluding the debris and the monocytes. Now we want to exclude let's exclude here the singlets, you know, in the same way. And so I'll do the same gate as I did previously, and something like that. So these are the single cells. Great. From the single cells, now I need to exclude the live dead, right? So exclude the dead, sorry. And now here I could put viability dye, and you can see here that I can already distinguish between them. You know, I could use Ford scatter, I could use site scatter, or I could even try and use other markers. And you can see here that I already see the dead cells that are positive, and I can see the CD3 positive. So I can gate already on the CD3 and do something like this. I could use a square if I'm able to let me call these CD3 positive. And then from here, I'm going to select CD4 and CD8. So let's say CD4 here, great separation. CD8, oh, not so much. Do you see here that we don't have any CD8 positives? Whereas when we were looking at the, let's see if I can go to that one now without losing too much time. This is one of the issues with FlowJo that we, oh, actually closed the wrong one. Do you see here the signal that we had with CD8? CD8 is labeled with APC compared to when we fixed it with ethanol. APC is a fluorescent protein. Most people don't know this, but PE and APC are fluorescent proteins that as Derek explained, they need to have water molecules. And so if you fix them with to be able to fluoresce, so if you fix them with ethanol, basically you dehydrate the surrounding of the fluorophore, and then it no longer fluoresces. So you're basically quenching the signal. And this is what happened. We were a bit lucky with the CD3, so the CD4 is FITSI, there's no problem. But with the CD3, we were actually relatively lucky, as you can see here. It's not as let me go back to this. Oh, here we can actually I can actually show you CD4 with CD3, and you can see that we did have a good separation with PFA. Not so pretty, not so nice with ethanol, but PE. So although PE lost some signal, we were still able to distinguish CD3. But you need to be very careful, right? If you use PE or APC, this is what you should expect. So if you don't have already a really good separation resolution, when you fix them with ethanol, you may lose completely, just like what happened with the CD8. So just be aware of that. And I think this was a great example. But again, we're gaining on CD4s, just like in the other sample. Let me go back here to the CD8, right? So we were getting on the CD4s and then looking at the cell cycle. Let's do the same here with the ethanol So these are our CD4s. And again, just for the sake of time, I'm going to be very quick and show you the cell cycle. So now we can go to histogram and then DAPI as a viability die. Now compare one and the other. I mean, just the, you can immediately see, and this is the same, the same scaling, and you can see you have way better CVs with ethanol fixed samples than with a PFA. And so there's always a balance, right? Ethanol is great for this reason, but then you have to be careful with the dyes that you use. So if you avoid any fluorescent proteins, you should be okay to use ethanol. If you do use, or if you have GFP cells or mCherry, etc., then the best approach would be to use PFA. And again, you should really optimize your protocol. If you're asking the question, okay, how would I analyze the cell cycle? This is the advantage of doing it in FlowJo. Now I could go, I could go here and say, okay, I have my population of CD4, and I can just hit here on cell cycle, and we would start playing around with the different models. So let's say I want to use the Watson pragmatic model to analyze cell cycle. We only have one peak, so FlowJo is having a hard time distinguishing the two peaks because we only have one. But if you play with a constraint, so let's say I'm going to constrain the range of G2, now it's able to just focus on the G1 and quantify G1 peak. Again, it wouldn't make much sense because it's all of it. It's all G1, or actually I should say G0, because cells are quiescent, they're not ready to divide. And so but this is how you would do if you had the normal DNA cell cycle profile. And I think that's it. I mean, in a very quick way. And I was going to show you how to, but we've already gone 15 minutes over. I was planning on showing you how to do the analysis with the raw data. You can do it with the raw data if you know what are the peak channels. Like I showed here, Kathy was showing the UV7 is the best peak for DAPI. FITC would probably be the B2 peak, and so you could actually plot on UV7, B2. I'll show very quickly before stopping. We could go to the raw, let's say to the raw ethanol. I would open this up, and I could, now I'm going to use just for fun. I'm going to use this gate. I'm going to use this gate, and then within this, I'm not going to now do the doublet exclusion, just for you to see. If I wanted to see, let's say that, so FITC would be blue 2, right? That would be the channel, and you see that I have here the CD4 T cells already, and what was it? CD3 was PE. So PE is the yellow-green one. I'm saying this, but I know already. If you don't know, you see, so we could analyze things without unmixing as long as you use fluors that don't overlap with each other, right? How do I know this? Again, you could go to the spectra viewer and you would just say, well, let's say PE to the Cytek spectra viewer, PE, and you can see that the highest peak here is in yellow-green 1, right? If I would look at FITC, I would see that the best peak for FITC would be B2, right? So, and you can even look at that in your own instrument, right? When you generate run the controls, the reference controls, you can actually see in that spectral plot which is the highest peak that you have, and use that one as your channel to do the analysis. So you could do it with the raw, or you could do it with the unmixed, depending on the type of fluors you choose. And that's it. I think I'm done with the analysis. I don't know if there are any questions.
Okay, actually, a couple just come through, Rui. Oh, you said that.
Oh,
So
okay.
actually, this might be down to Kathy or for Anna and Barbara if they're about. So were the cells rested overnight before they went to the staining after, or they just directly coming out of the freezer?
Right. No, I believe they were fresh. I believe, but Anna would have to correct me. Anna or Barbara would have to correct me. Actually, maybe I think they're online, so maybe they can add that to the chat and answer that.
Yeah, or even the the Q&A because they won't be able to participate in chat, I don't think, or can they?
Oh.
I think they might be able to say it just to us,
I
but
think they can.
okay.
And the other question is, can we use lower percent ethanol, lower than 70%, or lower incubation time for improving unwanted effects on that on PE? That's a very good question. Actually, Derek, do you have experience with this? Using
There's quite a narrow range. I mean, it doesn't have to be exactly 70%. I would say maybe between 60 and 80%. Actually, you'd think, well, surely 100% ethanol is better, right? 100's got to be better than 70. But that little bit of water that you dilute that in actually helps get the ethanol into all the phases of the cell, all the lipids within the cell. If you go below 60, then you don't get as good a fixation. So that will affect again the CV of the data. So there is a little bit of leeway, but I would keep it relatively close to 70%. And also with fixation, particularly with that ethanol, or any fixation, you should use cold fixative because fixation is an exothermic reaction. So we don't want to boil our cells. So we should always have cold fixative. Okay, do we have any other final questions? Otherwise, just a couple of slides to wrap up. I could share my screen.
Yeah, I'll stop sharing.
Which means I get to stop Rui sharing.
Mm-hmm.
There we go. Okay, so we've done that. Okay, so hopefully we've answered all your questions, and thanks everybody for the questions. There are some great ones there, and the whole purpose of these OpenFlow sessions is to try and make this as practical as possible. So it's been great to have those questions we've got along. I just wanted to put almost a final slide again, just to thank Anna and Barbara, as you can see from the photo there on the left, which was taken at the recent Cyto meeting. Anna's the only one who doesn't have a drinking problem, but we do get together occasionally, and it's great. In fact, this session was planned at the Cyto meeting, so it was great to get everybody together to be able to do that, and it's good to be back. So in terms of sort of going forward, what we'll do is we'll send a questionnaire to everybody, as we always do at the end of these sessions, because all your feedback is useful, because it tells us what you want. And if there's any particular type of application or type of machine or type of approach that you want us to look at, do let us know, because we will try and do that. Between us, we have access to all sorts of different machines, all sorts of different experience. And as we said a couple of times, this is being recorded, so we will make this video available via our OpenFlow cytometry channel on YouTube, and you can see the address there. And if you do have any comments or any questions afterwards, you can reach out to all of us via the openflow@gmail.com address, which will come to Rui and Kathy and myself. Hopefully somebody will answer. You can also, of course, comment on the YouTube videos. You can subscribe to the channel. You can like it as well. All those are very useful. And if you want to follow either me or Rui's Lab on Twitter, you are welcome to do so. So thank you, everybody. Again, thank you for staying till the end of this. We're not as good at timing as we could be, but we've got there in the end. So, yeah. Thank you all for coming. We don't have a date yet for our next session, but we will be carrying on this. I mean, there may be a summer break, but we will be back. Based on your feedback, we'll decide on what we're going to look at. So thanks everybody for coming. Have a enjoyable rest of the day wherever you are, and an enjoyable weekend. Hopefully the sun will be shining.
Yes. Thank you, everyone.
Thanks, everyone.
Bye-bye.
Bye, everyone.
Bye.